The Mpox virus is believed to have originated from a common ancestor within the orthopox virus family, maintained in small mammals, particularly rodents in Central and West Africa. Over time, the virus diverged into two genetically distinct clades based on genetic and phenotypic differences. The first clade is the Central Africa clade, also known as the Congo Basin clade. It exhibits a higher virulence with a mortality rate of about 10% and an increased potential for human-to-human transmission. Infections from this clade tend to be more severe compared to those from clade 2. It predominantly circulates in the Democratic Republic of Congo and surrounding East and Central Africa region. The other clade is called clade 2, which is also known as the West Africa clade. This clade is subdivided into two lineages, clade 2A and clade 2B. It generally causes less severe disease with a mean mortality rate of about 3.6% compared to 10% for clade 1. The current outbreaks, including the one that began in 2022, are primarily linked to clade 2, specifically lineages 2B. These clades reflect the virus's evolution adaptation to its varying impacts on human health, emphasizing the need for targeted public health response and vaccine development strategies to address the specific challenges posed by each clade. The evolution of Mpox clades, specifically the two main clades identified, that is clade 1, the Central African, and clade 2, the West African virus, is influenced by several factors. The first factor is genetic mutations. The Mpox virus has a double-stranded DNA genome that can undergo mutation. Although DNA viruses generally mutate slower than the RNA viruses, they can still exhibit rapid host adaptation through mechanisms such as gene duplication and the formation of gene accordions. These mutations can lead to variation in virulence and transmissibility between the clades. The other factor is zoonotic spillover. The initial emergence of Mpox virus in humans is linked to zoonotic spillover events primarily from animal reservoirs, which include rodents and possibly other wildlife. The interaction between human and these animal reservoirs can lead to the introduction of new viral variants into the human population. The third factor is human behavior and global travel. Increased international travel and changes in human behavior, such as the trade of exotic animals and close contact within special populations such as men who have sex with men, have facilitated the spread of Mpox virus beyond its traditional endemic regions in Africa. This could have contributed to the emergence of some of these new clades and variants we are currently seeing. The fourth factor is waning immunity. The cessation of routine smallpox vaccination after the disease's eradication in the 1980s has left a significant portion of the global population without immunity to orthopox viruses, including the Mpox virus. This lack of immunity can lead to increased susceptibility to infection and the potential for the virus to spread and evolve into new hosts. The fifth factor is geographic and ecological isolation. In Central Africa, particularly, the Congo Basin clade has evolved under distinct ecological conditions, including increased wildlife diversity as well as human-wildlife contact. These likely contributed to the virus's adaptation to more frequent transmission to humans. The sixth factor is viral adaptation itself. The ability of the Mpox virus to adapt to new hosts and environment can also be influenced by its genetic makeup generally, particularly the presence of genes that facilitate immune evasion. For instance, the West Africa clade has shown disruption in certain open reading frames that encode immune evasion genes, which may contribute to its lower virulence compared to the Central African clade. The evolution of Mpox Mpox virus clades is a complex interplay between genetic mutations, zoonotic spillovers, human behavior, immunity gaps, environmental changes, as well as viral adaptation. And understanding these factors is key for developing effective vaccines and prevention strategies against current and future Mpox outbreaks. More specifically, vaccines developed for smallpox provide cross-protection against Mpox virus due to the genetic similarities between the viruses. However, as the virus continues to evolve, there is concern that these vaccines may not provide equal protection against all clades, particularly if new mutations impact the virus's ability to evade immune responses in future. The emergence of these new variants within the West Africa clade and potential genetic drifts could reduce the effectiveness of existing vaccines. Thank you for watching, and see you in the next one.